So this is a case like where this type of simulation experiment actually drove not only my own research, but other people's research who had done this in the field and verified it in diverse germplasm of maize. So the simulation came first is what I want to point out. I've done a similar experiment in switchgrass, but we didn't cut away roots. This is one of the ones where we did rely on two different ecotypes of switchgrass, AP13 and VS16, if you're familiar with it. What I want to point out from that study is mainly that, first of all, that there's this lateral to axial ratio again. That's sort of like a relative, the higher it is, the more the relative allocation to lateral roots. Along this direction, without getting into the details, it's more or less like there's more stress going to the right. And so as there's more stress, the plants are allocating more to the lateral roots, and we can pick that up pretty readily. And then here, just to show like a direct relationship of root abundance to nitrogen uptake, I have a plot of the amount of 15N that was taken up by the shoot and the abundance of roots in that depth. So it kind of shows a direct relationship of root length with nitrogen uptake. For those of us who study roots a lot, the scanning method where you wash roots out of a sample, scan them on a flatbed scanner is one of the most common methods. I think of it as the bread and butter of root biology research. It just always works. And a lot of people have all these images and need a way to analyze them. I had been involved with several projects to do root image analysis. And eventually I decided that the computer scientists never really got the needs of the users right. So viewing myself as sort of the primary user of this software, while I was at Noble, where I had pretty good levels of core funding, which was really nice, we were able to develop a software that really does do what it says. It just works. You can download it from risevision.com. I think that we'll be able to continue its development now to try to improve it. But it's been used around the world. It's been downloaded more than 9,000 times. So I think that it's fairly successful. And I did a demo of it when I was at Washington State, and people are using it there, for example. So another aspect of my work is that, of course, I think a lot about where the roots are, how they're structured. But then when you study roots, you realize that a lot of people say that studying roots is hard, and they're just talking about doing that. But what's even harder is measuring what roots are doing. And of course, you all are getting at a lot of that here with exudation and stuff. You can almost turn some of your platforms on the head, though, right, to look at depletion of nutrients too, which you get at what I'm trying to show you here. So I mean, we know that there's nitrate transporters. We know that somehow they aggregate at the root segment level, and I think this is a big knowledge gap. We don't know things like how many transporters are there per area and why does it matter, and they aggregate at the root system level. So we wanted to understand more about this. So we developed a platform as well for doing high uptake, high throughput screens of uptake. Well, let's say medium throughput. I'll show on the next slide how that works. Some of these ideas might be relevant to how you all do things here. That we developed this kind of growth platform where we start seeds and put them in these little baskets with foam. We actually use these cone containers to put them into these common growth areas. And that's because if you just put the roots in there with a plug and try to pull them out later, the roots are all entangled. And so using these actually keeps the roots from entangling in the growth phase. And you want to take them out of the growth chambers to the measurement chambers. I designed this 24-channel system because it has peristaltic pumps on both sides. There would be other ways to do this type of thing too. This is pure hydroponics, where the pump on the left injects in the starting solution of known concentrations. The one on the right takes little subsamples and ran it through ion chromatography with both anions and cations. And so got most of the macro and micronutrients, not all, but most of them at once, which isn't very common. So we studied then you can convert that to uptake rates because we scan the roots and get their mass. So everything is standardized by root size instead of just net uptake. So we looked at the parents of the maize nested association mapping panel and could show that there's quite a lot of diversity for, here I'm only showing nitrate, but there's a lot of interesting results from the study about how uptake rates are correlated to each other and to root respiration. But this is also, it was heritable, which is a point for breeders. So there aren't very many studies that have shown that you could potentially directly breed for something like for uptake rate directly. So I think I've shown that roots are a major component of the plant's carbon economy. You can think about that in terms of architecture and kind of trying to make cheap roots. But another important aspect of carbon economy for roots is their respiration, because it's living tissue, so they're using carbon all the time. And so you want them to use carbon as efficiently as possible. But it's hard because you don't want too much respiration. But if there's zero respiration, it probably means it's dead, and you probably don't want dead roots either. So somewhere there's sort of a sweet spot in the middle between dead roots and ones that use too much carbon. And that's what I'm trying to figure out. So you can have like roots with lots of carbon dioxide being respired and roots with less being respired. So I developed a similar platform for that, and this is a wheat study. So this was actually my first genetic analysis that I did in my own group of wheat, working with our breeder at Noble. So we grew wheat in a similar type of way. Wheat's a bit smaller, you can fit more of these. We had them in these 15 milliliter tubes to keep the roots from getting messed up. I had a good team there, and we would go through and excise the roots and then put them into these chambers to measure CO2 flux and then scan the roots. And we also had like shoot mass and stuff. So we have 3 chambers going at once. So sometimes to increase the throughput, it's easiest just to do it in parallel and have more people there. If you can manage it, which can be hard. So we had these 3, and then for each sample, we have a text file with basically this type of information embedded. We have time and the molar concentration of CO2. So we developed and shared a script. Again, I'm very big into, I share all my R code, all my data. It's all available from my website. And the script will go through all the text files and fit this regression, pull out the flux, and then we divide that flux by the root measurements. And I'll just show here that the heritability, I could separate them by root classes like length and parent root, so we get kind of that lateral to axial root thing. And if we look at heritability, you can see that specific root respiration was heritable at 0.46, which means that most of the variation is caused by the genotype effect, which means it can be used for breeding. And at the same time, that specific root respiration, it's also interesting that specific root length, which measures the length of the root per dry mass, which means that you can build more roots with the same mass. So it's another carbon efficiency trait. Had the highest heritability, even higher than shoot biomass, which is somewhat surprising because we tend to think of root traits as being generally more variable than shoot traits. So one thing is that these are measured on the whole root system. So I would like to be able to partition really respiration into root types, like tips, the lateral length and the parent root length. But since it's on the whole root system and it's like really hard to like cut them apart and measure it independently, we can't do that. But what we can do is a statistical approach that I've been trying on various types of data. I think it's a useful approach where we fit a regression model to total flux based on the root tips, the number of root tips that rhizovision gives, lateral length and axial root length, and get a regression model from that. And then this is a plot of the predicted flux from the measured flux. And you can look at how the individual points deviate from this expected flux. And that's sort of how much respiration there is that's not caused by the root structure. So it's another way to think about differences in a trait. And I think it can be used in other ways. And it's also interesting to note in the study that it has direct evidence through the true measurements of specific root respiration versus shoot dry mass, where there's a negative correlation, which is what you would expect if it's true what I said, that there's sort of a wasteful amount of respiration, that you would see this type of negative correlation. So, I mean, I think if you look at a plot like that, you should be skeptical. So I acknowledge that there's a lot of spread in it, but at the same time, it is evidence of that line of thinking. So here's a real Manhattan plot like I showed you earlier with the cartoon. I promise you this is real data. And there's a threshold for where the significant p-value is essentially. And so we can basically, all the points above this basically end up being related to gene candidates that then people can follow up on. So specific root respiration itself in different forms had significant genetic regions associated with them. And so did other root traits like specific root length, average diameter, and root tissue density. And those are all kind of measures of carbon efficiency. And we also tried to push forward, like most of my data is multivariate. And most scientists just ignore the multivariate data and they still do univariate analysis. But in this case, we did do multivariate GWAS. And so these points in blue are from that. And you can see there's more statistical power when you do the multivariate traits. So it's useful. So I've shown you why root carbon efficiency matters with some of this work, I hope. And I want to just briefly thank my team at Noble from before I went to Oak Ridge, really great group to work with. And now I want to tell you more about when I went to Oak Ridge. You know, as you move through your career, you find yourself having to adjust to the times, sometimes reinvent yourself a little bit. But I took the opportunity to think more about my work. I actually have a PhD in ecology, so it's not really a stretch for me to think of ecosystems. I started trying to think about how this stuff gets put together and how it relates to the nitrogen cycle and to the carbon cycle with special attention to soil organic matter. And I haven't really, you know, accomplished all this or anything, but just sort of an aspiration to think about at least. So I worked a bit on, when I got to Oak Ridge, kind of trying to learn what's going on, what other people are doing. I'm the type of person that likes to collaborate and know what my colleagues are doing. So we know we have challenges to solve, like many modalities of root imaging that need different types of image analysis. A lot of it is time series. Sometimes we have these same type of images like you, where you have the roots as well as like the metabolites or other things, and how does that co-register and co-localize is a question. We have this FRED database already, the Fine Root Ecology database, and you know, how do we come up with common standards and share data efficiently? How do we scale the simulations? And so we have this group called the Digital Underground, where we're trying to increase interactions, collaborations among below ground researchers at ORNL. And of course, I could see how that would work across the national lab system as well. So some of the modalities of roots that I'm looking at now, of course, I have the bread and butter flatbed scanner of wash roots, which always works. I can guarantee you will get good results with it. And my website, risevision.com, not only can you download the software, but also has all the protocols for doing all these fairly detailed. So I think it's pretty useful to people. We also have a Rhizosphere on a chip platform that's still under active development and use. Jack Cahill developed a type of mass spec that is similar, I think, to some of the things here for targeting kind of a raster image of, in this case, amino acids. We have neutron radiography because we have the spallation neutron source, SNS and HIFER at Oak Ridge. And neutrons are very sensitive to hydrogen and water. It's pretty easy to segment the roots. These are kind of like 2D projections, but you can also do tomography. And Jeff Warren, who's actually in our environmental sciences division, has done some work to try to calculate things like water uptake from these. But again, to me, you know, how these, for example, these two type of things relate to each other is how I see like the needs for generalizing image analysis, co-registration across layers of imaging data, and looking at how things are influencing each other across the layers. So I think there's some people doing that here that'd be interested to learn more. We also have mini rhizotrons in the field where you put clear tubes drilled into the soil in the field, and you can put a camera down it and get images. And it's the type of thing that people have done over time, but then they have like thousands of images they never analyze because they realize it's a lot easier to get the image than it is to analyze it. So over time, I hope to start to address some of these. So I want to talk a little bit about our plant microbe interfaces, SFA. So I've been involved with for just a couple of years, but it's been ongoing for many more than that since 2010. We just had our latest annual meeting most recently. You have a alumnus here with Eastland. So we're thinking about how does populus, the tree species, influence the microbiome around it, largely, basically. But recently, I've been kind of tasked to try to understand some of the imaging needs and analysis needs in it. So at the lab level, we have things like the rest chip. We have like the fungal chips that Eastland worked on, did really nice work with. We have lots of different styles of plate imaging, looking at cold cultures and things like that. Some fluorescent imaging, and more cold cultures. At the greenhouse level, we have our advanced plant phenotyping laboratory, which I'll talk to you more about in a little bit. I'm also working more with rhizoboxes. And at the field level, we do have field level experiments, but not very many explicit field level types of measurements, but things like UAV and satellite analysis would be relevant. And we do have people like in our environmental sciences division that work on that. So with rhizochips, me and each land try to find ways to image them, because I was especially interested in like the root side of it. It's really hard when you start to do all the microscopy and stuff. But if you just want to see the root structure, it's not so hard to get good images of that. And I worked on some methods to try to easily segment out the roots. So with rhizovision itself, it's not that the strength of it is not in segmentation, because that's a really hard problem. So we focused on trait measurements. But you can use other software upstream of rhizovision, which there's actually several independent examples of that now in the literature that it works. Here, this is a UNet that did the segmentation. And as kind of proof of concepts, I was able to get kind of time series data for roots. And then again, thinking about this multivariate aspect. So we have several traits and time. How do you analyze that? I have that as well with our advanced plant phenotyping lab as well. So with rhizoboxes, a rhizobox is basically a thin box, and the roots are growing in 2D, typically grow at an angle. So the roots are gravitropic, so they hit the glass and kind of spread out. Not all the roots, but like some proportion of them. And if you can get good images, then we can automate the analysis. So this is the rack. I was telling Amir about this earlier. This is like sort of the rack system, and the rhizoboxes that I've made that have handles, so it's meant that the users can easily take them in and out. It's not fully automated, but it's like a smart way, for people to do the work efficiently. Once we get the images of the roots, we have we have different options. We have a new staff scientist who focuses. It's John Lagergren. I don't know if you knew him. Yeah, he started as a staff scientist to focus on AI and math. He can do this type of stuff pretty quickly of implementing new types of tools. So we segment the roots, and then with rhizovision, we can analyze them. Over the next year, I'm hoping to create a headless one for rhizovision where we can call it from a script instead of having to use the user interface. So we can try to increase the throughput. So one small example is I have a colleague in environmental sciences division, Verity Salmon, who wants to look at how peat soil causes waterlogging and how the roots grow and how methane gets put out. So they had these rhizoboxes. So I helped kind of set up like a way to measure them that's pretty good. Like, it's not too much glare. These systems can have a lot of glare. And even just by itself, this is segmentation only from rhizovision, that it could do like this. And so it's not perfect, but I think it means that with a little bit more advanced methods, we can do much better segmentation. This last summer, I had a GEM. I don't know if you have that internship program available here, but it's an internship. And Shanae Otley from North Carolina State worked on. So sometimes when we scan roots, there's too many roots to fit in the scan. So we split them up across scans, and then there's not really been a standard method for how do you put the data kind of back together. So we've come up with two different ways. One way is to concatenate the images. So you just make one giant image, and then you can run that through rhizovision. That works just fine, but requires a Python script. The other way is an R-based method where I'm trying to show here is that we have the ID here, NNN, means it's the same sample. So you have NNN, OOO, then all A's. So those letters are how the images should group. And so these were individually run through rhizovision. So each row is for one of the subsamples. And then I've developed a statistical method to where you get the aggregates of all your traits. Sometimes that's just adding them up, which is, even I can do that. Sometimes it's more complex, like how you need to put it back together. And we took the concatenation as sort of the ground truth, and then compared it to the statistical aggregation. And these are the type of plots you see. So I have 3 image sets from different colleagues to show that it's kind of applicable across systems. The regression models here, it's always like a good fit. But for some things like length, it's a nearly perfect fit, which is what you would expect because adding it together. Some of them are more sensitive because like things like a median, you can't really recreate statistically after you've already done the aggregation for an image. so they don't fit quite as well, but it's typically pretty close. So we're planning to release this to the community so that they have options for how to handle this. Because what I had seen, when I literally walked into the office and like a postdoc will be sitting there like with like Excel open trying to group one, like just linked together to get the additions, and then what happens is that people, because they only knew how to do links, that they're ignoring all the other data. But like I said, I tend to think of everything as multivariate, and so I want to make sure that we kind of keep the multivariate features through the pipeline. So here's an example of popular roots. This was 10 scans concatenated together, so it's 81,000 by 10,000 pixels, which is 800 megapixels, which I think is a little less than some of the Samsung phones now. Not really, but Samsung is pretty crazy. And what I want to ask you is what's the length of roots in these 10 images? Each one's about the size of a sheet of paper. Does anyone want to guess how many meters of roots there are here? You don't get a prize or anything.
Kilometer, I would say.
A kilometer?
Yeah.
Not a bad guess.
100.
A little less.
All right.
187. So it is a lot. And if you had like the root hairs or something, it would be like way over a kilometer. So one of the things that I've worked with each land that we would like to figure out more ways to do it is with the hyphal growth. And I'm not sure if there's like a proper name for these, but I sometimes call it a microchip. These, is that good? Yeah. Microfluidic devices with really small channels that plant hyphae can grow through. And she was looking at different structures. So here's the pipeline we had in place then. Now that we have more of like a specialist to handle this area, I think we'll be able to make more progress. So here's just an example. She had these different types of chips, like some with more of a grid structure and some with more of a pore structure. And then you can look at how it changes sort of the velocity front of the hyphae growing through. I know that she's noticed a lot of interesting things about how hyphae get stuck in channels and circle around. We won't really be able to get to those kind of local level measurements very soon, but I think that the global measurements of which channels are filled and which pores are filled, we'll be able to start getting at more over time. And again, it's multivariate time series data, so we're trying to figure out how to handle that. I want to tell you a little bit about our advanced plant phenotyping laboratory, APPL, we call it Apple. So it's a system where this whole thing, I should have a picture of the facility so you can see the scale, but this is a fairly large greenhouse area where the plants are growing on a conveyor system. A little bit bigger than this room, maybe like almost twice as long as this room, maybe, or about that or something. So it's 500 pots essentially, or you can have trays with more than one plant. And they're conveyed through the system into this brick building that was built, which was when they did the project, building a brick building was the bulk of the cost. The plants move through several imaging stations, which I'll explain on the next slide. So they move through and get imaged, the data stored, and they move back out, and then they go out to the back where there's like a watering station and things like that. So the type of imaging that we do, there's chlorophyll fluorescence, which relies on putting out strobing of one wavelength of basically red light, and then having a camera filter to measure the fluorescent light. We can get measurements of the efficiency of photosystem 2. We have color images from the top and the side, which is basically what's on your phone. We have 2 types of hyperspectral. There's V-near, which covers 380 nanometers to 900, and SWIR, shortwave infrared, that goes from 900 nanometers to 1700 nanometers. We have a thermal camera that measures the leaf temperature. And that's important because as plants use water and water leaves the leaves through transpiration, it cools the leaf down. So you can get some idea about water use from that. And we have a 3D laser scanner that delivers a point cloud. So the vendor of this system, PSI, supplies some analysis. I won't go into the details of the table. They supply some analysis, but then in parallel, we're developing our own image analysis. We've had a lot of kind of a nucleation of image analysis team recently. So I think over the next year, we'll be able to start tackling some of these challenges that have been ongoing for years, really. Also with data handling. I'd be really curious to hear how you all manage all your vast sets of data. I was just hearing about the MAUDI. I know it's like a terabyte per scan, probably the X-rays, the CTs, like a few gigabytes and stuff per scan. So it adds up, you know, and like how you keep track of stuff is hard, especially as you analyze it. How do you keep track of what you did, who has the data, like what should happen next? So yeah, there's lots of features we get, like lots of spectral signatures, plant size, leaf area. Like I said, like things like quantum yield from photosynthesis. We have some cases like where we have to do data and interpolation, but we have started now to start doing these genetic analyses where we can discover significant genetic regions for hundreds of traits. So then it's really more of a problem, like how do you prioritize gene candidates after that, really? Now getting to gene candidates is easy, but we can script all this. The GWALs are fairly intensive analyses, like they take a few minutes on a laptop, but we can do them in parallel on our cluster computers, no problem. So I wanted to tell you about another experiment in Apple from Plant Microbe interfaces SFA that the PI is Dave Weston with a postdoc, June Lee, who's done really nice work. So this is looking at, we did Arabidopsis and poplar, and this is the results for Arabidopsis, but it's a bacteria called Vorax. That's why we do these kind of inoculated versus non-inoculated studies. We've been trying to learn ways to do that in the greenhouse, and also with soil inoculations more broadly, like trying out different methods. That's a really interesting area. But so you have these ones that are inoculated versus not, and then they're in 3 different temperatures. And then we're looking at some of the effects. So here's some of the biomass effects and things like that. Now, one of the first things that we get from Apple now, one of the things that I'm starting to deliver to our customers as sort of like a summary report are things like a heat map of the correlations among all the traits. Because like I told you, like as a physiologist, this is kind of the first thing that I look at. And like a geek like me will go through this and like look at it, like, oh, how does this trait relate to this one? And it's really useful. It's sort of like the first thing you do to look through this. Now, in a group like this, I don't expect you to see the individual traits and stuff, but I did color code the modality. So you have chlorophyll fluorescence, RGB, SWIR, and VNIR, hyperspectral. And what I want to point out really is that if you look across this band of CF, you'll see that large blue dots means there's a large positive correlation, basically. And you can see, I think, even from the audience, that even as you go across different modalities, it maintains different types of correlations. So it's interesting to me that the modalities can predict each other. And what does that mean physiologically is the type of question that a person like me would be interested in. But then, of course, the biologist just want to know, like, was there effect of the bacteria? Please tell me there is. But yeah, we make no promises. So, and one of the things that's interesting, actually, we measure size in different modalities. So some of the strongest blue circles across here are when it's a measurement of size, and they give like the same result. That's good for us as like a data check. It's important to kind of have like a quality check that's built in. You can see that here, like how size this is from color to chlorophyll fluorescence, and from hyperspectral to chlorophyll fluorescence. They're very good fits. One thing jumped out to me was that there was this kind of nonlinear correlation, but a nonlinear really relationship between VNIR and quantum yield from chlorophyll fluorescence. And at first I didn't have this color coded, so I just saw these like 2 distinct groups. But then when I looked into it, it's that we can either, for chlorophyll fluorescence, you can dark adapt the plants where they don't get light so that all the electrons like go out. And I'm not a biophysicist, but that's essentially what happens. And then you can have them light adapted where you take the measurements after they're in light. And so those are what's distinct. And then asking, this is NDVI, which is a vegetation index of kind of how healthy our green plants are. So getting into things like how you handle the multivariate data, this is very sensitive to picking up the time points. So you can pick out plant age quite easily, not only from size though, but also from like the spectral signatures. But then when we color code by like circles and triangles, that doesn't pull out as much here. Then again, PCA is not meant to really do clustering. It's not really for clustering, despite that people almost always show it as if it is. But it just shows you the maximum components with variation, with no knowledge of the factors involved. And also one of the things that we're delivering to our customers is that for all the traits, like for every trait, you basically get plots of the data. That's the most useful place to start from. Usually is kind of visualize it, right? you might have seen some examples of like people who have data sets where they never actually look at the data and they draw these conclusions, but then if you plot the data, it looks like a bear and stuff like that. I've seen these type of examples from our like stats people. So they get all these plots, you can start to look through them. One of the ones that we noticed was chlorophyll fluorescence, and that's what kind of the focus of this was. That you can see a separation by quantum yield at lower temperatures. At higher temperatures, though, is interesting because you only got that separation at the first time value, but then that actually makes sense because we know that variovorax, the red is the variovorax with the bacteria, increased quantum yield, which means that it's helpful. But we know that variovorax basically confers stress tolerance that otherwise the plant develops by itself over time. So it makes sense that's the first time point we see that difference. And then it's also interesting that this is the type of trait that we wouldn't usually measure, but there's this big difference in leaf shape. So I have to follow up on what's causing this. But that's at the later time points at the high temperature. So just briefly, I wanted to tell you about my journal, Plant Direct. I'm editor-in-chief. It's a journal published by the Society for Experimental Biology as well as the American Society of Plant Biologists. We do both direct submissions. That's most of the submissions are direct, but we also accept transfers with reviews from our partner journals like Plant Physiology, the Plant Journal, the Plant Cell, and Journal of Experimental Botany. So if you're ever publishing a plant science paper, you can submit directly to us, which I'd love. But you can also think about strategies like submitting to one of our partner journals and then allowing it to be transferred to us to make a fast evaluation. With these transfers, we can usually make a decision within two weeks. So with that, I want to acknowledge the people whose work I've shown, and of course, Oak Ridge Department of Energy, Center for Bioenergy Innovation. And I'm really pleased to be here. If you have any questions, please feel free to ask them now or later.
Questions? Just simple question. You show the variation of the nitrate uptake rate difference in different genotypes of maize.
Yeah.
Did you see any significant correlation with their phenotypes? So polar or greener or more products or something like that?
Yeah. Among root phenotypes, especially, like we had correlations with among nutrient uptake rates, which isn't really known, like it's not, so you can see that there's like a positive correlation of like phosphate uptake with nitrate. And if you ask people like which would make sense, you'll hear both answers. So like it's not necessarily expected that they should be correlated because they have different transporter. Also with nitrate uptake and respiration, root respiration. Among the shoot traits, actually, it's interesting. We did very detailed analysis of the shoots, and we've been working on like a second paper that kind of looks at this like whole plant trait coordination, like the type of thing you're getting at. And I don't think there's necessarily like these are fairly young plants. It wasn't really a relationship to shoot size, but I think there was to shoot in content. We did nitrogen content, the shoots and the roots. But mostly I think the main conclusion will be the way that we'll probably tell the story, whatever, is that there's sort of like a root syndrome of traits and a shoot one that's independent, kind of.
Yeah, go for it.
Can you say a little bit more about any results from the neutron studies, if you have any interesting findings?
For the neutrons, like I said, that's a colleague, Jeff Warren. I'm still wanting to apply for time on it. I think so. I mean, they've published one paper fairly recently and maybe like one or two in the past about it. But I think that the conclusions are mostly, things about like where water is taken up compared to like the root tips and things like that.
Yeah,
But if you look up Jeff Warren, you can find his work.
I figured there will be some interesting following the water flux.
That's the idea. If you have it like over time, you can actually get. And some other people, there's a person, a soil physicist named Andre Carminati, who has done work like that as well.
It will also be interesting to correlate it with X-ray and then you see different things.
Yeah, definitely.
Thanks, Larry. That very impressive body of work. I had a question about your respiration results. I don't know if I fully understood them. So is the takeaway that you can kind of predict like 40 to 60% of the variation in respiration by having a bunch of the structural traits?
I didn't think about it like that. Yeah, like, you don't need to measure it for something.
Well, that's,
Yeah.
I'm trying to avoid measuring.
That's a very valid point. Yeah, I didn't think about that. No, like what I was trying to say is that you can't predict a lot of it, but you can't predict all of it. And
Okay.
the part you can't predict is the interesting part. Because it's like, there's more respiration, the way I have thought about it is that there's more respiration than you would think, only based on the architecture. And that's because there's some intrinsic difference in like maybe like some of them, so we discovered gene candidates and so like some of them were related to ATP. But unfortunately, my past institution like laid us all off. And so I was working with like a plant breeder to look at stuff like that. But since we all had to leave, I don't think I'll ever really get to go back to that data set because it's in wheat. But I am hoping that people who study, I was trying to talk to them about some of the people at Washington State, because you all had, on my way out, I saw plenty of winter wheat on your Palouse hills coming out here. But maybe they'll take up some of the gene candidates. Yeah.
But I mean, 60% is still not bad. I guess it's better than no respiration data, right? So if you can,
Yeah, but that was total flux. I mean, yeah, you expect like, I mean, like total flux should definitely relate to like the abundance of roots. But then like that part that, so I mean, just thinking about carbon economy, yeah, like we know that more root volume, like more root mass is more carbon expenditure. And you expect like there to be a comparable amount of metabolism related to it. So you're right that like it is, you already kind of know that just based on like the root abundance. But what's really interesting by measuring the respiration is that you get like the differences in what they're really using. And so for me, it's still the really cool thing about respiration. It's actually one of the fastest root measurements to take. It's faster to take root respiration than it is to scan roots and stuff. It's faster than doing the nutrient uptake. So it's actually a pretty, to me, a useful method.
Any questions from people on site? I mean, online, virtual folks? Don't see any.
Something in the chat, maybe?
I'm not looking at chat. There's no question. Just a quick follow-up on the respiration. So have you tried or how do you see if the respiration traits that you're measuring, as far as I understood, have different root zones, and you see different responses from different root zones regarding their respiration? How that could be correlated with some type of rhizosphere traits and rhizosphere functions?
Yeah, well, I think that for sure. Yeah.
For example, exudation profiles of those specific zones, or how that shaped the microbial community. Have you ever looked at that?
I haven't. I could make some guesses that I better right. One of them would be that I know that root tips have higher root respiration, and we also know that root tips are the center for exudation and stuff. And then you would expect like more rhizosphere community there. But yeah, I think that type of question would be like really interesting to look at more.
Very cool. I have one comment online.
I think that was just about muting. The one from.
Were they telling me to mute?
No, no.
No, no, you. All right.
Here's the follow-up on
It's a tough crowd.
Like the respiration study was done in hydroponics, right?
Yeah.
So if I understand correctly, respiration versus investing the carbon for respiration versus the root mass production, like the cell wall production, like the biomass, forming new root structures. Investing the carbon for respiration versus the new cell wall production or the biomass production.
You mean like the result, like why is the respiration? I mean,
Yeah.
The respiration is really broke down into different things. There's growth, which would be getting into like cell walls. There's also, I mean, the roots are very active. So there's a lot of production of ATP and like mitochondrial respiration directly from that. And a lot of that is for nutrient uptake because most of the transporters require a ATPase proton pump, and the transporters are symporters with protons.
Right.
So a lot of the respirations for that.
Yeah. So if you could have done that in the soil instead of hydroponics, do you expect that to reflect much better respiration readout. Like what I'm trying to say is in the hydroponics.
They will not, yeah, I think that we should always ask if we would get the same results in a lab in the field. And I think the answer is almost always no. And then the question is like, is it useful information?
Yeah.
And then that's like what we may not know. One thing with like field results is that sometimes people talk about like lab to field doesn't correlate, but then like I look at field data, I'm like, well, actually field to field data doesn't correlate. So is it really a lab to field question? Or is it just like a question of plants are very plastic. But with root stuff, clearly growing in soil will make them different. So one thing you would want to know would be like, does the relative ranking, like the respiration might change, but does the relative ranking of the genotypes stay the same? Maybe then it's kind of like, doesn't matter how much it changes. But we also know, hydroponics can change the root structure, like less root hairs, things like that as well. Some of the stuff I picked out like that statistical technique allowed me to say that root tips had about 40 times the respiration. According to that model, like if you take into account the root tip number, it's 40 times the respiration. And I think that stuff like that would be the same, that you would still find that root tips have the most respiration. And, so some things would be kind of like stay the same and some things could be different. That difference is important. And for all of us that do lab and field work, we have to keep, questioning things like that.
Right.
Yeah.
Just a couple of comments.
Is there a time for one more?
Oh, sorry, please.
Thanks for a very interesting presentation. It might be a silly question, but what are the processes behind more allocation to roots with stress?
Yeah, so we do know that in general, it seems like in times of stress, there's like a kind of a crude trait called shoot to root ratio that's been used for a long time. Where you just get the dry mass of both. And shoot to root ratio tends to go down, which means that there's more root allocation with many types of stresses. And the question of why is interesting. I was talking to the people at Washington State about that with like whether plants are sort of more driven by source or sink limitations, like that's really like an active area. In some cases, like if you increase the sink strength, then all of a sudden like the source or photosynthesis rates can go up too. But it seems to be that and those also change during the developmental stage. But yeah, there's definitely things like sugar transporters and how plants respond to sugars that seem to guide that type of allocation.
One last thing here, and this might be naive too, but a lot of what you talked about is the benefit to the plant for the nutrient uptake, and I get that as the main driver. But in times of stress, and this may be what you've already had with some conversations from Amir and others, is there some way that you now measure or can put in what's going out, to interact with the microbial community? So not just uptake, but the other. And so does that lead to more diversification of lateral roots, for instance, or something like that, at that point where you're now expending carbon, but maybe to get something back?
Yeah.
And is that, do you see that already in your data, or is that something?
I have not studied exudation myself till now, but we have people at other people at Oak Ridge who are interested in it, and I think I'll definitely be doing more in that area. And definitely, last year my early career award was based around. I didn't get it. My proposal was based on sort of like whole plant carbon economy and trying to do this type of phenomics approach, but then including things like exudation as well to have sort of a total plant carbon budget.
Great.
But this year the call is for microbiome, so I can't just like reuse that.
Cool.
I think it's really interesting. It needs to be answered. Yeah.